everyone, my name is Janna and I'm a PhD candidate with the Johns Hopkins score in sex and age differences in immunity to influenza. Today I'll be talking about how sex, age, and frailty interact to impact the humoral response to seasonal influenza vaccines in older adults. Older adults bear the highest burden of severe disease and mortality associated with influenza, and annual vaccination is the best method of prevention. To this end, two vaccines have been specifically developed for older adults: the adjuvanted vaccine and the high-dose vaccine, which contains 4 times the amount of antigen as standard vaccines, and which will be the focus of my presentation today. Despite these vaccines being specifically developed for older adults, efficacy remains an issue. The efficacy of the high-dose vaccine was estimated at only 32% for the 2015 to 17 seasons. In addition to poor efficacy, large heterogeneity in the immune responses mounted to the vaccine have been observed in this population, with some individuals mounting very poor responses. Our research focuses on how host factors may explain both the heterogeneity and the poor efficacy observed. Specifically, we are interested in how sex, age, and frailty interact to impact the vaccine response in older adults. To address this hypothesis, we work with a longitudinal cohort study that began in 2014, so today I'll be showing you 6 years of data. Each year, screening begins over the summer and focuses on community-dwelling older adults above the age of 75 in the Baltimore area. Those who are enrolled in the study are assessed for frailty using the Frailty phenotype, which uses the signs of shrinking, weakness, slowness, exhaustion, and low levels of physical activity to classify participants as robust, pre-frail, or frail. Serum and peripheral blood mononuclear cells are collected to measure baseline immunity, and then the participants are vaccinated with the high-dose vaccine. This vaccine targets 3 strains of influenza, but today I will only focus on the immune response to the H1N1 strain. 28 days after vaccination, there is a second biological collection, and the main outcome of the study is the hemagglutination inhibition, or HI titer, which is used to assess seroconversion status. Individuals who mount a 4-fold or greater rise in antibody titers comparing day 28 to day 0 are said to have seroconverted. So let's dive into the results. First, we looked at the effects of age and frailty separately. Here I plotted the estimated probability of seroconversion against age on the left and frailty on the right. These probabilities were estimated from adjusted logistic regression models separately for males and for females. If we start on the left, we see that in females, in purple, the probability of seroconverting decreases significantly with age. In males, however, in green, while there is a similar trend, the decrease with age does not reach statistical significance. Moving to the right side of the slide, here we see that among females, there is a significant increase in the probability of seroconverting as frailty increases. Once again, however, the association seen in females does not hold true in males, where the probability of seroconverting does not change significantly with frailty. Next, instead of looking at age and frailty separately, we looked at the intersectional effects of these 2 host factors. Here again, I plotted the estimated probability of seroconversion from our adjusted logistic regression models against age. On the left, we are looking at this association only among the robust or non-frail participants. As indicated by the p-values beneath the graph, in this group, there is no significant association between the vaccine response and age for either males or females. This means that if you're not frail, age does not seem to impact your ability to mount a response to the vaccine. On the right, on the other hand, among our participants who do show signs of frailty, we see the decrease in probability of seroconverting with age again, but this trend is only significant among females. In conclusion, coming back to our 3 host factors of interest, we saw that age and frailty both interact with sex, since the vaccine response only decreases with age and increases with frailty among females. At the intersection of all 3 factors, we see that the vaccine response only decreases with age among females who show signs of frailty. Together, this data demonstrates that host factors can be used to explain some of the heterogeneity observed in vaccine responses among older adults. It also shows that it is insufficient to look at host factors in isolation, as aggregating data can mask the more interesting trends that we see in the population. Understanding which older adults are able to mount strong responses to the influenza vaccine and which are not is an important step in addressing the continued burden of influenza in this population. We hope that these findings can ultimately be leveraged to design better vaccines or vaccination strategies for this population. With that, I'd like to thank Dr. Sabra Klein, Rosemary Morgan, and Sean Lang, and all of the other people that were involved in this project. Thank you.